Hello, thanks everyone for joining our KOL discussion on gene editing with Dr. Jennifer Doudna. I'm Gula Lifshitz, a biotech analyst at Chardan, as you probably know by now. Dr. Doudna likely doesn't need much of an introduction for this audience. She's a professor at UC Berkeley, a co-founder of numerous companies, including several that are presenting at this conference, and has been a pioneer in the genome editing space with respect to the basic science, as well as in translational applications, particularly for CRISPR nucleases. So in this session, we'll talk about how the genome editing technologies and their applications have evolved and where the field goes from here. And yes, so I'd like to again thank Dr. Doudna for participating, and maybe we can start by you telling us a little bit of a brief overview of CRISPR technology and its origin story before we dive into more deep discussion.
Sounds great, and thanks so much for inviting me. It's a real pleasure to be here. CRISPR is a technology for gene editing, as I think many folks are aware, and but you may be less aware of where it came from. So it's a technology that originated with curiosity-driven research into a bacterial immune system that uses a programmable set of proteins to find and destroy viruses. And what makes CRISPR so good as a technology for genome editing is that fundamentally it works by finding particular DNA sequences and cutting them. And the enzymes that do this, which are called CRISPR-Cas or CAS enzymes, these Cas proteins are directed to DNA sequences by molecules of RNA that provide the zip code, the sequence information to find a particular site in a DNA, whether that DNA is a small piece of DNA in the laboratory or whether it's an entire human genome. And so scientists over the last 8 years have been able to harness this for targeted cleavage of DNA in cells that triggers site-specific DNA repair, and that's where the editing happens. So that results in changes to DNA that can be as small as a single base pair in the human genome or a plant genome, or very large changes that delete entire genes or sets of genes and replace them with other genetic information. So it's a very powerful tool and has really opened the door to lots of opportunities that I'm sure we'll get into.
Fantastic. So yeah, since the initial reports of the CRISPR editing in about 2012, the technology has been studied pretty extensively by thousands of academic groups as well as industry groups. So from your perspective, what are some of the challenges and limitations that have emerged from some of the initial studies of first-gen CRISPR nucleases?
I think what's amazing about these CRISPR proteins, nucleases, is that they work robustly in many different cell types, and that's what has really made the technology take off the way it has. That being said, the fundamental the way that they work is to trigger DNA repair, and that's where the editing happens. And that's also where either off-target editing or editing that can't necessarily be controlled by the scientist can also occur. And so I think one of the current challenges in the field is that it's relatively straightforward to make genetic knockouts of genes, disruptions to genes in many cells. And in many cases, that's the kind of application that one desires. But making changes to DNA that are more subtle in a way, such as altering a DNA sequence to correct a disease-causing mutation or introduce new sets of genes, those changes are trickier to control. And so that's one of the ways the field is developing, is really to try to find ways to use the technology with more, I would call it precision for those kinds of targeted changes. And then of course, there's all the challenges that probably many people here are familiar with. For any kind of genetic manipulation, one wants to ensure that those manipulations are accurate, that they're safe, that they're effective at making the desired changes to the genome that are indicated for a particular application. And I'm sure we'll get into this as well, but I think these are all areas where there's been a lot of work and a lot of research in CRISPR, but there's still more to be done.
Absolutely. So over the past several years, I think there have been a number of efforts from your group and others to characterize some of the novel nucleases, for instance, isolated from different bacterial species. Can you explain this approach and how some of these newer enzymes could expand the capabilities beyond the current state of the space?
What's exciting to me about CRISPR is that it's a family of enzymes. It's not one type of protein, it's a whole set. Nature has evolved these to deal with many different kinds of viruses in bacterial cultures. And so in the laboratory, it's possible to harness many of these for genome editing applications. Why do this? You might say, well, as academics, we're curious about the whole sort of CRISPR biology, and we're trying to understand it, and that's certainly true. But I think as a technology, it's important to have many of these proteins available for applications because, as we're finding out, there are particular properties of different enzymes that might be more or less important or relevant given the specific application. And I'll give you an example. So one of the current challenges in the field for biomedical uses of genome editing is how to deliver editing enzymes into cells or into tissues. And doing that there are different ways to do that right now, but one of them involves packaging the genetic material encoding the enzyme or even the enzyme itself into either a viral capsid or into some kind of a nanoparticle, where we find that the smaller the enzyme, the better for packaging. And so finding and developing some of the smaller versions of CRISPR-Cas proteins for genome editing, I think could be very useful. We think a lot about the chemistry of these systems in terms of the properties of these proteins that might be more or less amenable to crossing cell membranes, as well as being active in different kinds of cell types. And the sort of early days, I would say, in that right now, but I think the early evidence suggests that having different types of CRISPR proteins available for uses in different cell types will open the door to more effective applications, just because some of these enzymes, for reasons we maybe don't fully understand yet, work better in particular cell types than others.
Mm-hmm. Great. And so those are the natural variants, but then there's this other effort to use engineering or directed evolution to further enhance the functionality of known nucleases. So how do you think about this strategy, and could it become complementary to the novel nuclease discovery efforts?
I'm really excited about these engineered nucleases. I think it's a really interesting aspect of the field that what's being done is fundamentally to rely on the mechanism of DNA recognition that these CRISPR-Cas proteins naturally utilize, which is an RNA-guided system, but then to build into the proteins additional capabilities that, at least for now, we don't find in nature. So an example of that would be being able to make a chemical change to a particular nucleotide in DNA that doesn't require cutting the DNA first. And that's a technology called base editing, and can be done using engineered forms of CRISPR-Cas proteins. And another example is using CRISPR-Cas proteins in a fashion that doesn't make any direct alteration to the DNA sequence in cells, but instead alters the way that genes are expressed, that could lead to changes in the output of genes in a targeted way. And those changes are called epigenetic changes. And so CRISPR can be used for this as well. And I think that's an area where there's a lot of future opportunity, because it opens the door to non-permanent changes to genomes that will nonetheless have beneficial outcomes.
Great. And so those are all approaches focusing on the nuclease component. How much innovation has there been, or is there likely to be, on the guide component of the CRISPR system?
Well, I think there's room for a lot more work there. The guide component, as you're alluding to, is the part of the system that provides the targeting information. So it provides the sequence match that has to occur for these CRISPR proteins to be active. And there's been some engineering of these RNAs, certainly beginning with our work with Emmanuel Charpentier to make the guide a single piece of RNA that could be used as a way to direct Cas9 to its target sequences in cells. And then since then, various groups have looked into ways to put either, fluorescent fluorophores, ways of detecting RNAs in the cells, and also ways of introducing molecules that could be useful in gene editing. For example, sequences of DNA that can be brought in through their interactions with the guide RNA that might be then used in cells for genome repair that could introduce new sequences during the course of editing. And I think there's more room for development there. It's an area where that's RNA is near and dear to my heart as a research topic, but many scientists who are working in the field of gene editing, I think, are naturally more familiar with working with proteins and manipulating those, and we've seen most of the focus there. But in the future, I think that changing the guide sequence and the guide structure may provide additional opportunities for engineering that haven't yet been explored.
Mm-hmm. Great. So I guess from a big picture standpoint, what do you think are the parameters that are important to consider when determining whether a nuclease has the potential to move from being a great scientific tool into therapeutic applications?
Boy, that's the big question, and I wish I had a pithy answer for you. We find that a lot of these proteins are active initially in the laboratory, but it's not necessarily trivial to turn them into robust genome editing tools. That being said, I think there is we've seen a pretty good correspondence between biochemical activities and what ultimately turns out to be useful in cells. And so when we see in our biochemical experiments, we see enzymes that appear to be quite active as DNA targeting or DNA cleaving proteins, or even RNA targeting enzymes, then we're encouraged to pursue those for gene editing applications as well. And we often find that we need to make various kinds of changes to these proteins to get them to work efficiently in cells, whether it's figuring out how best to deliver them into cells, or as you alluded to in the previous question, how to make sure that the guide RNA is being actively made and associating with these proteins in cells where that's essential for the gene editing function to occur. And then there's some elements that are sometimes hard to predict. For example, finding very small versions of CRISPR-Cas proteins that one might have, just by looking at a sequence, thought that they might lack some of the capabilities that are found in larger proteins. And yet, when we test these both biochemically and in cells, we find they're remarkably active and effective at gene editing. So we're still trying to understand why that is, but that seems to be a sort of an empirical observation.
Great. So I think as an analyst covering the gene editing space, I think one of the most common questions that I get is about comparing nucleases or platforms. How do you think, so from your perspective, how do you think about questions like, is this nuclease more specific than that nuclease and the like? Do you think there's value to these type of general efficiency or specificity benchmarks, or is it more important to consider kind of an individual guide or target?
Probably more the latter. I mean, I think my overall answer is it depends, because that really does seem to be the case. That we find that with these different types of proteins, they have different activity levels in cells that are not necessarily easy to predict a priori, and they also have different requirements for guide RNAs that can affect the way they function in cells. That just has to be sort of figured out empirically. That being said, I think that at least in our experience, we've seen that there are quite a few now different types of CRISPR-Cas nucleases that are highly effective for genome editing. And to me, that bodes well for a future where we have an increasing, expanding toolbox of proteins that have different gene editing capabilities, and one will be able to select the tool that is most useful for an application based on its properties. And in the commercial sector, of course, providing enough CRISPR-Cas proteins to go around, so there's not a bottleneck at the level of FTO, and that lots of companies will be able to get access to tools that are effective for their applications of interest.
Mm-hmm. And so from that perspective, I guess as a follow-up, if you look at an asset that's already in development with a particular nuclease technology, how do you think about that relative to the evolution of the space?
I'm not quite sure I understand the question. I mean, I think you're asking, if you have a set of different enzymes with maybe different properties, and they also have different sort of patents around them, and is this sort of
no, just around, let's say, if there's a company that has something with SP-Cas9, how do you think about an asset that's already in development with SP-Cas9 relative to potential future development of similar assets but with a new technology? If something already works, is that good enough, or do you think there's likely to be an improvement over moving to a new nuclease tech?
It's tricky, isn't it? Because, I think what I've seen now in a number of different studies and groups, and this is both in academic labs and in companies, is that, you might have a technology, you might have an enzyme that works pretty well for something, for a gene editing application, let's say. And so you're moving forward and heading towards the clinic with it. And then something comes along that is, marginally better or has some property to it that might be advantageous. Do you go back and redo all of your work with this new protein, or do you just carry on? And the answer is that at some level we try to do both. So I'll give you an example. So we have a pretty active program in sickle cell disease going on at the Innovative Genomics Institute right now that I'm involved in, and we're heading towards the clinic with that. And but frankly, all of our data, our IND enabling data, are with an early version of CRISPR-Cas that was developed, really going back a few years now. And we know that there are now improvements to that technology, where there are things that we could do that would make the enzyme more efficient at editing. And so we're trying to decide, what do we do? And so we're kind of doing both. We're still heading towards the clinic with the early technology, but we have a research team that are continuing to develop newer versions of the enzyme, so that at some point, if we get to a point where either we run into an issue in the clinic, or we decide that the new technology is just so much improved that it's worth going back, that we will have that option.
Great. And so maybe coming back to some of the parameters that could differentiate across different nucleases and or are important things to consider with nucleases, perhaps. How do you think about immunogenicity to different nucleases? Is this an issue? What are the challenges associated with that?
Yeah, there's been a lot of discussion about this, as you know, in the field and in the media. And I think that I have a couple of comments on this. One is that these are bacterial proteins, so they're clearly foreign proteins, and so it's not surprising that they would trigger an immune response, either due to exposure to the types of bacteria that these enzymes come from. If they're pathogenic bacteria, humans may have been exposed to them, and we know that there is evidence for some low levels of antibodies to CRISPR-Cas enzymes that one can detect for that reason. And then you could also imagine that administering these proteins therapeutically could also trigger potentially a larger immune response that might be problematic in certainly for repeat applications of the enzyme. And so how do we deal with this? Well, I think in a few ways. One is that there's a lot of fundamental research going on right now to figure out just how big of a problem is this. And I think the field still doesn't really know the answer to that question. And so we're working with David Rollet and Greg Barton, who are two terrific immunologists at Berkeley, on a DARPA-funded project right now to understand immunogenicity in mice and see if we can mitigate an immune response to CRISPR-Cas proteins by making changes to the surface of these proteins that might make them not as likely to induce an immune reaction. So that's one kind of strategy that can be taken. And then, there's also the, going back to what we talked about previously about the fact that in nature, there are many different versions of these CRISPR-Cas proteins. And I think that's actually an advantage, right? Because it means that we have many shots on goal. We have ways to, use these proteins where you could imagine as if you did have to use it for repeated treatments of a patient, that you could do it with different versions of CRISPR-Cas that would not be cross-reactive in an immune sense. And then, of course, there's lots of knowledge in the field from working over the years with biologics, with antibodies, et cetera, that companies have developed ways to make them either humanized or sort of resistant in different ways to immune reactions that I think could be applied to CRISPR-Cas proteins as well.
Mm-hmm. Great. That makes sense. And so I guess some of the other parameters, kind of in the nitty-gritty science part of the technology that might differentiate across the different nucleases include kind of the way that they cut, whether it's blunt versus staggered, and whether they have similar or different ability for targeted insertion. So how do you think about those? Do you see differentiation across the nucleases for those parameters?
I do. I think that's a really interesting possibility. Nobody's yet really shown robustly that the type of cleavage of DNA that happens in cells is really, significantly impacting the way that editing occurs, but it could be. It still remains to be, I think, very rigorously analyzed. But that being said, certainly you could imagine that, having different efficiencies of DNA recognition, different accuracies of DNA recognition, and then just things like cleavage kinetics, can all really affect the outcomes of editing. We found recently in a study that we were doing with the Broad Institute, actually, that that having an enzyme that is very, very fast at sort of sliding on and off DNA can actually be disadvantageous. It might sound like something that would be a good property, but it actually, in some settings, can actually trigger more off-target editing than one would like. And so, you might want to actually have an enzyme that is slower at that, or, find variants of that starting enzyme that you can dial back the kinetics of DNA binding to a point where you don't observe a lot of off-target editing. So those are the kinds of things that we're now getting into the weeds a little bit here, but I think, having real mechanistic understanding of these proteins is going to be very helpful.
Mm-hmm. Absolutely. As you say, I guess it's still in the early stages for some of these detailed studies on the variety of nucleases that are available. So I guess from that standpoint, do you think specific nucleases will emerge as winners for particular applications, for instance, in vivo versus cell therapy or targeted integration, or is there a value to having broader portfolios? How do you think about that?
I still think there's value in having a broader portfolio, but that being said, I do think that what we're starting to see is that for a particular enzyme, you can develop a whole suite of sort of widgets and applications based on that foundational protein that can be used in a variety of different applications. And so, this has happened certainly with the original Cas9 protein that was, that Emmanuel Charpentier and I worked on and published in 2012. And then subsequently, we've seen this with some of the Cas12 proteins. And now with these smaller enzymes, Cas-X, Cas5 are a couple of examples where teams are taking those proteins and their fundamental properties and then just adding things onto them to give them different capabilities, but still relying on the fundamental properties of the original enzyme.
So you touched on this a little bit earlier, but for in vivo editing applications, the field has really leveraged some of the delivery technologies that had been developed for gene therapy and RNAi, such as AAV vectors and lipid nanoparticle technologies. So from your perspective, how is delivery shaping the applications of genome editing, and how might this change in coming years as delivery technology evolves?
I think up until now, what's happened is that a lot of the applications that we're seeing clinically are in systems or tissues or cell types where editing is, where delivery is challenging. And so that would be for cell-based therapies where editing can be done ex vivo and then cells delivered back to a patient, or in tissues like the liver or the eye where there's an opportunity for delivery just due to the location of the tissue or its natural function, like in the case of the liver. And so that's been very exciting, of course, for the field to see those advances happening quite rapidly. And I think what's on the horizon now are opportunities in other diseases that would require, strategies for delivery that would be, systemic in a patient. For example, thinking about treating a disease like muscular dystrophy, where you would need to deliver to muscle cells and do that in a systemic fashion. I think, certainly the laboratory data and even animal data so far look very exciting there. And now, the field is at a point where how do we take those early results and turn it into a strategy for treating people where we need to be able to deliver systemically and safely, and avoiding immune responses and other kinds of toxicities, ensuring accuracy of editing, all of these things that come up in that are more complicated when you're doing systemic delivery, because you don't have the opportunity to verify editing the way you do in, say, ex vivo types of applications. But that being said, I think that's very much on the horizon. I think over the next, say, 5 years, we're going to see real advances in those types of applications of CRISPR as well.
Mm-hmm. Yeah, and you mentioned some of the smaller nucleases, the Cas5, Cas-X. Can you talk a little bit about those and how they fit into the landscape?
Yeah, these are interesting because they are naturally smaller enzymes. And so they have different chemical properties. They have, just aside from just being physically smaller, so potentially easier to package into a particle, whether it's a virus or some other kind of, lipid nanoparticle, to also just fundamentally the chemistry of these proteins is different. But we think there are some differences in the way they actually interact with DNA. The structures of these proteins are distinct from some of the larger enzymes. And that's interesting because it opens the door to additional engineering opportunities to add other functionalities that might be harder to apply or to append to very big enzymes because they're already very large. If you have a smaller platform that you're starting with, then you have a way to add additional functionalities without making the protein so large that it's either very difficult to express in cells or package and introduce into cells. So I think, time will tell. I mean, I think these are very interesting systems. They, some of them, in the case of the Cas5 enzyme that you mentioned, are found, to our knowledge, entirely in viruses. So they're carried around in viruses that infect bacteria. So these are obviously have very tiny genomes and naturally, for that reason, probably have to have really compact proteins as well in these CRISPR systems. So they're naturally compact. They are, at least in our hands so far, highly active and effective at triggering DNA cutting. And so there's the potential there to take a system that started off, with the original enzymes being quite large proteins, and now working with proteins that are, half or less of the size, and just all of the, opportunities for delivery that I think that opens the door to.
Mm-hmm. Yeah, I was going to ask about other potential advantages to these beyond delivery, but I think you've already answered that in terms of the ease of the engineering, other kind of additions to them. Is there any other advantages beyond the size for these? Particularly if we think about non-viral delivery, where the size constraints are a little bit less rigid.
Yeah, I think just thinking about the particle size for a smaller protein is obviously more compact. Also, some of them have, very small guide RNAs that they use. And this gets back to your earlier question about engineering the guide and thinking about ways to modify it. If you have a very small, very compact guide RNA, then again, there's the potential to make changes to that could be advantageous and without sort of introducing a lot of additional material that has to be packaged and delivered into cells.
Great. So maybe shifting gears a little bit, you mentioned this earlier, but obviously safety and specificity are at the forefront as the use of the genome editing in humans expands. So what is your perspective on how to think about issues, not just like off-target editing, but also some of these large on-target deletions and rearrangements that have been seen, which might not be the central focus of specificity assessments?
Yeah, really important work that has uncovered these and shown that they can happen. I think that's essential that we know the potential for this. That being said, I think that there are some really effective ways to limit the occurrence of those kinds of genetic rearrangements that largely include minimizing the activity of these enzymes. And that might sound contrary to what you would think, right? You would think, well, I want the most active protein. I want the best genome editor. But it turns out that I think in some applications, at least, it turns out to be better to have proteins that are have lower levels, at least, of biochemical activity, because it does also limit the likelihood of triggering, multiple cleavage events in DNA that could result in these kinds of genetic transformations. So that's one thing. And then also, I think just, knowing that these can occur is important, because it does now give scientists a toolbox for detecting them and making sure that we have ways of mitigating against those kinds of rearrangements, starting with just knowing their occurrence and their frequency. So that's now a type of assay that's, I think, done standard type of assay for any cell therapy applications of genome editing.
Mm-hmm. So maybe just as a quick follow-up to that, from what you've seen, how standardized are some of these assays to assess specificity and other issues that might arise with gene editing?
Well, I wish they were more standardized. Looking in the scientific literature, it certainly, often seems like we're comparing apples and oranges when we look at data from different studies. However, over time, I think there is emerging kind of a set of standards in the field where people are using various types of, I would call them sort of capture-seek kinds of experiments. There's lots of different versions of this, but ways that one can detect off-target editing and comprehensively identify those sites in cells through a sequencing-based approach. I think that's become a very standard strategy in the field, and maybe the details of how that's done might be different in different studies, but it's become a real expectation, I think, in most studies that that type of analysis would be done. So that's good. The National Institute of Standards and Technology, NIST, has been for several years now has convened a team of scientists coming from both industry and academia to develop a kind of a standard set of assays for detection of editing accuracy in particular that I think has been useful. And I don't think they're done with that, but I think it's a really important endeavor, because we do need to have a series of standards, not only in terms of the way we detect editing outcomes, but also the way we think about them. So for example, if you do an experiment, you do an accuracy assay and you see 0.1% off-target editing overall in the genome, do we worry about that? Do we worry about it? Is it dangerous or not? Right? I think there's been a lot of focus on ensuring that editing is absolutely 100% accurate, but in a way that might kind of be the wrong question, because maybe what we should be asking is, are we seeing off-target editing at sites that matter in the genome? And if we don't, then maybe that's not where we should be focusing a huge amount of effort and resources. So I think that question is still very much in sort of discussion in the field, and I hope will be addressed by this NIST group as well.
Yeah, absolutely. And so I guess in terms of safety, is there potential for some safety signals to derail the progress in the space? And how can the field learn from maybe some of the safety issues that were seen in the early gene therapy trials?
Yeah, I think about this a lot. I think that the history of gene therapy is fraught. And I think many are familiar with the death of Jesse Gelsinger and how that really set the whole field back at least a decade. And so, for those of us now working with gene editing and the CRISPR technology, I think there's a real desire to ensure that although we all want to see the technology advance as quickly as possible, we also want to make sure that it's moving forward safely, and that safety is always the number one concern. And it's challenging. I mean, I think there's lots of trials that are either underway or planned, and so this is going to increasingly expose larger numbers of people to the technology, and with that kind of exposure, there is risk there. I think for many of us, there was real excitement in the last year with the announcements from a couple of groups, the company CRISPR Therapeutics, as well as the group of Carl June, that had announcements about phase 1 trial data showing that CRISPR seems to be safe in human patients. I think that was very, I think I sort of heard a collective sigh of relief when those early results were announced, even though they didn't get to the question of efficacy yet, but it really just saying, look, at least in these early trials with initial small numbers of patients, we're not seeing signs of toxicity or rejection that would be red flags for going forward. So this is, I think, cautious good news in the field, and I just think that we need to continue to collectively proceed with that level of caution.
Mm-hmm. Great. And so maybe again shifting gears to additional applications or technologies within the CRISPR space. You mentioned earlier some of these approaches like base editing or fusion of epigenetic enzymes to CRISPRs. Can you talk a little bit about where you see the potential there and how we should think about those technologies?
I'm pretty excited about that whole area of development because it does open the door to new ways to use CRISPR that are, just again, provide more opportunities for applications. In the case of base editing, it's a way that one can make chemical changes to DNA sequences without inducing a double-stranded DNA break, which, as we discussed previously, could be problematic if it's leading to genetic rearrangements. And so the potential to make a targeted chemical change to a DNA base is, it still blows my mind to think that one could actually do that in a genome. It's just extraordinary. Is the technology there yet? I would say it's not, right? I think, if you look, down in the read the fine print, so to speak, you find out that, well, in fact, there's a lot of editing that's going on that's sort of off target with the use of base editing that still needs to be dealt with. But nonetheless, I think the promise of that technology is certainly interesting and has attracted a lot of work and attention in the field, rightfully so, because there is real opportunity there. Do I think it'll be possible to get to base editors that have real precision? I think it is, I really think it is. And I'm a big believer in, continuing to really understand mechanistically how these proteins are working so that their properties can be controlled to enable that kind of precision. And then we talked about, a little bit about these epigenetic editing forms of CRISPR. And there, again, it's an opportunity to use the fundamental CRISPR platform not for making a permanent chemical change to the DNA by cleavage and repair, but by making a change to its modification state, maybe its methylation state that triggers all changes to genetic output. I think that's also really, really interesting and exciting. It hasn't been explored as much outside of the laboratory yet, in the sense that I think right now those kinds of epigenetic modifying forms of CRISPR have been largely used for, genome-wide screening and things like that in cultured cells in labs, where there's lots of really cool science coming out of it, but not yet sort of real evidence of clinical utility. That being said, I think the field might be on the verge of that, because we're getting to a point where it's possible to really control the activities of those proteins. They're getting easier to manipulate. And there's some evidence now in mouse cells, at least, that these epigenetic modifications can persist over multiple generations of cells, which is extraordinary. It's sort of really, really interesting, and has, I think, real potential for clinical use in the future if that holds up.
Mm-hmm. Yeah, so I guess that would place these epigenetic modifiers somewhere between the DNA editing and the RNA editing that you also mentioned earlier.
Exactly, yeah.
And
right.
So the RNA editing, that's also kind of not a permanent modification. So from your perspective, where do you see that fitting in?
Well, I think that's more along the lines of what people have done with, RNA interference, where, it's possible to manipulate the transcriptome, transcripts in a targeted fashion. So far with these CRISPR enzymes, because of the way they work with RNA targeting, they're not that specific. So I think there's still a lot of, just fundamental work to be done to turn them into a tool that would provide the kind of specificity for transcriptome, editing that would be needed certainly for clinical use. I think right now they're still in the realm of, sort of an R&D effort and using them for research studies. Time will tell whether they can be engineered to have the kind of specificity that would be desirable for the, sort of real genetic manipulation, the way we have seen with RNAi.
Mm-hmm. So I guess in this last section, maybe let's look forward. And so for the moment, what do you see as the remaining questions and challenges in the CRISPR space that might be answered in the next several years?
Well, we didn't talk too much about integration of new DNA, right? And that's something where there's a lot of promise for genome editing, but it hasn't been realized yet, I would say. And why is that? Well, it's harder to do that, and it's harder to control DNA repair that triggers that kind of insertion of new genetic information. Do I think it's possible to get there? I do. I think it's will it happen in 5 years? I'm not sure, honestly. I think what we're likely to see is probably relatively incremental improvements. That's sort of what we've been observing over the last 8 years as various groups have worked on this. Now, is it possible that there will be some, breakthrough, a new enzyme that has some kind of, targeting, DNA insertion capabilities? Yeah, absolutely. I mean, one development along those lines was the discovery of these CRISPR transposases, right? That can actually, insert DNA into genomes. The challenge there is they don't do that scarlessly, right? They leave their own DNA signature when they do integration, and also so far they haven't worked in any plant or animal cells. They're working in bacteria only, so they may end up being of more limited use in that regard. But nonetheless, I think that's an area of real development. And then I also think that, we talked about this already, but, the whole issue of delivery is still very much goes hand in hand with CRISPR, because to really open the door to genome editing in a broader sense than we're seeing today is going to require some, I think, real innovation on delivering gene editing molecules into cells and tissues, especially to be able to do that systemically in ways that are safe and efficient. So that's an area where there's, again, a lot of active work, but I think it's really at the forefront of the field currently.
Mm-hmm. Yeah, so maybe this ties into what we just talked about, but those are some of the challenges. Are there particular areas where you think we are likely to make considerable progress in the next few years?
Well, I certainly think, these ongoing clinical trials, we're going to see outcomes there that will, really show the efficiency and the effectiveness of genome editing, in particular for, the kinds of cell sort of immunotherapy advances that are being made in cancer treatments. and also for treating all kinds of blood disorders that involve genetic manipulation. I think that we're likely to see some really significant advances on those fronts in the next 5 years for sure. I'm hopeful that we'll also see some real progress in treating certain types of genetic diseases. And the two that come to mind, well, maybe three that come to mind for me are muscular dystrophy, cystic fibrosis, and Huntington's disease. So these are all going to require, these are all, monogenetic diseases, very well characterized biology, and particular patient groups that are affected by these diseases. And in each case, we know we have laboratory data showing that CRISPR can absolutely be effective at correcting the underlying disease-causing mutation in the lab. And so now we want to, I think the field as a whole is really moving towards taking those laboratory results and figuring out how to turn them into a robust therapy, again, requiring effective delivery, safety, evaluation, and getting into a lot of the questions we talked about earlier about the immune response and the persistence of the outcomes of an editing type of therapy. Will it really be effective over the course of a lifetime, especially if you were to use it in a very young patient, such as a boy affected with muscular dystrophy? Would this kind of therapy then be a one and done treatment that would persist over the course of his life? So, these are all the kinds of things that are being investigated currently in animal models, and I think we're likely to see, real progress in the next 5 years.
And then kind of looking forward even more, is there anything that you think that sounds like sci-fi now that might become feasible or common in the next 5 to 10 years?
Personally, I would really like to see CRISPR become a standard of care for certain types of disease. And the one that I think about the most right now probably is sickle cell disease, because I think that's, we've already seen the results of, initial clinical trial data from the one patient in particular, Victoria Gray, whose sickle cell was effectively cured using CRISPR. I think that's, very exciting. However, it's a $2 million treatment right now. And so going from where we are today to turning it into an actual standard of care where a patient affected by this disease can receive a treatment is going to require a lot of things. Reducing cost is high on the list there. And right now, it kind of sounds like sci-fi to be able to deliver a treatment like that as truly a one and done therapy without requiring bone marrow transplantation. Mm-hmm. And I think that, I'm really excited about the potential to do exactly that. In other words, come up with a delivery vehicle that would be so safe and effective that you could give it to a patient like Victoria Gray and have it hone in to the cells that need editing, leave the other tissues alone, and do its job and then go away, so that the patient is left with corrected genes and basically a cure for their disease. I mean, this would just be extraordinary. And if one could do it in a disease like sickle cell, then I think that will create a model for how we can do it for other rare diseases, and maybe even a way to use CRISPR to its full potential as a truly personalized kind of therapy. And again, that'll require, I can't really quite imagine it today, how we're going to deal with regulation and, all of the clinical trial requirements that are, you clearly wouldn't be able to do full-blown clinical trials for every iteration of CRISPR to treat each individual patient. So we do have to come up with models that will ensure safety, but also ensure that the technology can actually move forward. So that's, another thing that's kind of, fantasy today, but I think is something really, really worth grappling with because it has such major potential.
Mm-hmm. Fantastic. So I see we're just about at the end of the session. So I really want to thank you again, Dr. Doudna, for participating and for the great discussion. And yeah, it was great.
Yeah, really great to talk to you, and a pleasure to be here.